This is a screencast describing the central dogma of biology. The central dogma of biology describes the flow of genetic information in biological systems from DNA to RNA and eventually to proteins. To better understand this dogma, it is important to understand what DNA actually is. DNA, or deoxyribonucleic acid, is the hereditary material present in humans and almost all other organisms. DNA stores information in the form of genes, which are essentially the chemical makeup of your entire body. This biological information is stored in the form of 4 chemical bases: adenine, thymine, guanine, and cytosine. These 4 bases pair up with each other, adenine with thymine and guanine with cytosine, to form molecules called base pairs. Each base pair is also attached to a phosphate and sugar molecule. Together, a sugar, phosphate, and base molecule form a nucleotide. As depicted here, nucleotides are arranged in long strands that will eventually form a double helix. The double helix's structure is somewhat like a ladder, with the sugar and phosphate molecules forming the ladder's walls, and the base pairs forming the ladder's rungs. The central dogma of DNA describes the way in which genes are expressed in the form of proteins. As I stated before, your DNA is responsible for containing the genes that will later be expressed as proteins. However, your DNA cannot leave the nucleus. This is because there are several chemicals and enzymes in the cytoplasm that would degrade and destroy the DNA, leaving cells unable to function. However, genetic material still needs to reach the ribosomes of the cell in order to be expressed as proteins. That's where RNA comes in. An analogy that's helpful in trying to understand the relationship between DNA and RNA is to think of DNA as the reference books in a library. You can never take these reference books out, however they contain vital information that you will need to use later. You can come into the library and take notes on the reference books, and take them out of the library to use for other purposes. Your RNA is analogous to these notes. As I just explained, RNA is necessary for gene expression to occur. The process by which messenger RNA, or mRNA, is created is referred to as transcription. In this process, a DNA strand, referred to as the antisense strand, is copied into the form of RNA, which is referred to as the RNA transcript. Structurally, mRNA is extremely similar to DNA. However, while DNA contains the base pairs adenine, guanine, cytosine, thymine, and guanine, RNA contains the base pairs guanine, adenine, cytosine, and uracil instead of thymine. The base uracil forms a base pair with adenine. There are 3 main steps in transcription. The first one is when RNA polymerase binds to the antisense strand on DNA. RNA polymerase knows where to bind due to an element called the promoter sequence in DNA, which essentially is a marker for the RNA polymerase to bind to. The second step in RNA transcription is referred to as elongation. Certain proteins, referred to as transcription factors, unwind the DNA, enabling mRNA to copy only a single strand of DNA, as I stated before, which is referred to as the antisense strand. The third step of transcription is referred to as termination. RNA polymerase continues to move along the antisense strand of DNA until it reaches something called the termination site, which is a specific sequence of DNA that tells the RNA polymerase to detach and disassociate from the DNA. Because translation occurs in the ribosomes located in the cytoplasm of the cell, the mRNA transcript must cross the nuclear membrane. However, before the mRNA reaches the ribosome, several steps must occur to prepare the mRNA transcript. Eukaryotic DNA contains regions of DNA that are not coded into proteins and are just not expressed. Currently, we do not know the purpose of these regions of DNA. These are referred to as introns. These are intervening sequences that must be removed from the mRNA transcript through a process called splicing. The remaining portions of mRNA that are translated into proteins are referred to as exons because they are expressed. Once the introns have been removed from the mRNA transcript, the remaining mRNA transcript is referred to as a mature mRNA transcript. This entire process of splicing mRNA is carried out by small protein complexes referred to as spliceosomes. After splicing has occurred, one last thing must happen to the mRNA transcript. In order to avoid being degraded by enzymes and chemicals in the cytoplasm, the mRNA transcript must receive a poly-A tail and guanine cap in order to receive protection from the enzymes in the cytoplasm. Translation is the final step in the central dogma of biology. The mRNA transcript is integral to this process. Every 3 bases of an mRNA transcript is referred to as a codon. Each codon then matches up with a specific anticodon carrying an amino acid. The structure of a tRNA molecule is depicted here. This portion of the diagram is the mRNA codon, which matches up with a complementary tRNA anticodon. This tRNA anticodon then contains an amino acid. Every tRNA anticodon of the same sequence must contain the same amino acid. Only different tRNA anticodons contain different amino acids. Before translation can begin, the ribosome must be assembled from 2 ribosomal subunits. These subunits contain attachment sites for tRNA molecules. They consist of the E site, the A site, and the P site. During translation, the mRNA will move through the ribosome until it reaches something referred to as a start codon. At this point, a tRNA molecule carrying an amino acid that is complementary to the mRNA codon will bind to this starting codon. This will begin the process of translation. As mRNA moves through the ribosome, each codon will be matched up with a complementary anticodon, and a chain of amino acids will start to be constructed. When tRNA moves from the P site to the A site, they are released. This allows the tRNA to rebind with an amino acid and enter the translation process again. This whole process will continue until the mRNA reaches a sequence referred to as the stop codon. The stop codon is bound to a release factor which causes the ribosome and mRNA to dissociate. This marks the end of the process of translation. At this point, several tRNA molecules have passed through the ribosome and created a peptide chain. This is the formation of a protein. However, simply because translation is done doesn't mean that the creation of a protein is finished. The protein must first be folded and modified. Sequences of amino acids in a protein determine how it will fold. Folding may also require the assistance of other proteins referred to as chaperone proteins. Additionally, a protein may need to be chemically modified before it becomes functional. For example, some proteins require the attachment of a carbohydrate chain in order to function. In some cases, some amino acids may need to be removed, and in others, the polypeptide may need to be shortened in order to become functional. Once this process is complete, a protein has officially been created through gene expression. That concludes this screencast on the central dogma of biology.